So I'm really fortunate for the opportunity to bring this panel together for this exciting topic and have an opportunity to present in a little bit of a different format, but at least bring people together for the scientific conversation that we hope to have as part of this panel that we put together, myself and the co-chair, Dr. Carolyn Menard, for this symposium for Society of Biological Psychiatry. The topic is Molecular and Circuit Mechanisms of Inflammation and Depression, Translational Research and Treatment Opportunities. And the idea really was to bring together both clinical and basic researchers. So as you'll see, we have four excellent talks, and it's not an accident that two of them are basic scientists and two of them are clinical scientists, but all focused on this question of depression, mechanisms of depression, and the role of inflammation. So I'm very excited to get into it. The way myself and Caroline will run it together, and we'll introduce each speaker as we go. We will leave time for panelists to ask each other questions. And at the end, we hope to have a discussion among all the panelists on this exciting topic. So with that said, let me introduce our first speaker, who is also, as I mentioned, co-chair, Dr. Carolyn Menard. I have my notes here. So Carolyn is assistant professor of psychiatry and neuroscience. She's faculty of medicine at the University of Lava and the Cerveau Brain Research Center. I got to know Carolyn when she was at Mount Sinai as a postdoc with Scott Russo, where she was involved in multiple projects regarding neurobiology of stress, inflammation, and depression. Interestingly, she's also worked in industry in R&D, and she currently holds something called the Sentinel North Research Chair on Neurobiology and Stress and Resilience at her institution. And I'm delighted that she's co-chairing this with us and looking forward to her talk. So Caroline?
Okay, so I'm going to introduce you some work that I've done while I was a postdoc in Scott's lab at Mount Sinai, but also some work, a paper that was published earlier this year from my own lab. So I've been a faculty for 2 years, so it's kind of exciting and new research for us. So I'm going to talk to you about social stress, and then I'm going to talk about the brain, but also the vasculature of the brain, so the blood-brain barrier, and a little bit of inflammation. As James mentioned, it's basic science, but we validate what we find in mice, in human tissue, so you're going to have a little bit of clinical data as well. And so in my lab, we are interested in depression, but we take a different angle. We not only look at neurons, but we also look at other cell types, including endothelial cell of the blood-brain barrier. Why? So the prevalence of major depression is about 7% throughout a year, 20% throughout lifetime. It's now the leading cause of disability worldwide. And then one thing that's unfortunate, it's 30 to 50% of the patients are resistant to current antidepressant treatment, which leads us to think that maybe some of the neuron-centric therapies are ignoring some causal biological mechanism. And so that's why we are interested in the neurovasculature and inflammation. Why there's some clinical findings suggesting and showing that the comorbidity of cardiovascular disease, depression goes from 7% to 17-27% in those people. And if you have a stroke, there's 50% chance that within the next 5 years, you're gonna develop some depression. So in those patients, it's often chronic and recurrent. So we think there's some common mechanism underlying these changes, and two of them could be inflammation and vascular dysfunction. So how do we see that is basically if we start on the left, so I'm going to use my arrow. So if you start on the left, we think under chronic stress condition or in depression, you're going to have some changes in the periphery. So you're going to have some organs are going to produce immune cells. Those immune cells are going to be trafficking into the bloodstream, and then they're going to be recruited into the brain. And so they're going to affect the brain eventually by, this is the part we are studying here, so the blood-brain barrier. Eventually, they're going to damage the blood-brain barrier, open it a little bit, and then it's going to change the cell on the other side. So the astrocytes, which are lining the blood vessel, but as well as microglia, which Georgia is going to talk to you a little bit about next, and eventually the neurons and the circuits. So you're going to affect behaviors. And so one question I often get, especially in clinical settings, is how do you know a mouse is depressed? So you have on the left the constellation of symptoms associated with depression in human, and you have in red everything that we can measure in mice. So we have a pretty good idea if a mouse is happy or not. And so one thing I'm going to talk to you a lot about today is isolation, social isolation, and avoidance. And so one thing we know is in human as well as in animal, chronic stress is the main risk factor to become depressed. And so we take advantage of that to study the biology behind depression and resilience. And so the animal model we're using mostly we use in Scott's lab when I was a postdoc and also in my own lab, it's chronic social defeat stress. So it's kind of a model of bullying. It's going to induce depression-like behavior such as social avoidance, anhedonia, anxiety. It's reversed by chronic, this phenotype is reversed by chronic but not acute antidepressant treatment, a bit like what is observed in human with the common antidepressant. And what's really interesting is it's a model of depression but also resilience. So the way it works is basically we have these two strains of mice, the regular B6, which is the nice one, doesn't bite fingers, so the student love it. And then so we expose them to a larger male, aggressive, that's a retired breeder, so they are more territorial, they're gonna be more defending their home. And so they are exposed to each other for 5 to 10 minutes per day physically, and then they're gonna be housed in the same cage. You can see on the right, it's an hamster cage with a divider. It's transparent, a plastic divider with holes so they can smell each other. So you have a sensory stress over the remaining 24 hours. So we do this for 10 days. So they see the B6 is going to see a new CD1 mouse every day for 10 days. After that, at the end, the following day, on day 11, we perform a social interaction test. So the way it works is we put the B6 in the arena. They are allowed to freely explore. There's a little interaction zone we define in green here. So the first time there's no mouse in it. And so the B6 can freely explore. And then after 2.5 minutes, we remove them. We put a new CD1 white mouse they never saw before. And then we see if they're going to keep interacting. So we define, I'm going to talk a bit about it later in the next few slides, the SI ratio, so social interaction ratio. So it's the time spent in the green interaction zone when the aggressor is present divided by absent. So you get kind of two subpopulations of mice. You're going to have the one that become, we say, depressed. They're stress susceptible, they show depression-like behavior. So that's the one at the bottom. The SI ratio is going to be below 1. So they are stress susceptible. They're going to avoid the social target. But then you also have a subpopulation of mice that are considered resilient. So they're going to keep interacting with the CD1 they never saw before. Mice are a social animal like humans, so they're going to want to see a new mouse. Maybe it's going to be a friend or not. And so when we do a bunch of mice, we get a distribution of about 2/3 that will become susceptible or depressed, and then 1/3 is going to be considered resilient. So it allows us to study the biology of depression, but resilience as well. And so what I did when I was in Scott's lab is we tested blood-brain barrier permeability. So we knew that in patients who are depressed and resistant to antidepressant treatment, they often have higher inflammation in the blood, and we were wondering how this inflammation circulating in the blood could affect the brain. And so there's this barrier in between, the blood-brain barrier, that generally is going to maintain impermeability to the brain, so it's hard to cross. And so what we observe in our mice after chronic social defeat, the 10 days, we screen them, and then we subject them to MRI scanning. And so what we did is we inject gadolinium. We did a baseline scan, and then inject gadolinium to test the integrity of the blood-brain barrier. So gadolinium is really small, and so it's used in clinics in human as well. And so what we observe, we look at different brain regions. The one I'm going to talk to you most today about is nucleus accumbens. So it's involved in mood regulation, reward, and different emotional valence mechanism. And so you have on the bottom left, see the behavior of the mice. So 10 mice that were on stress control, so they were just housed in regular cages, never exposed to the aggressor. 8 mice that were considered stress susceptible, and then 12 that were considered resilient. So when we test the blood-brain barrier integrity, what we found, the way it worked, the heat map, so if you have higher infiltration of the gadolinium, so loss of blood-brain barrier integrity, it's going in toward yellow. If you have a maintenance of the blood-brain barrier integrity, it's gonna go below, so it's going to be going toward blue. And so what we observe, if you look here in the NAC that I highlighted, in stress-susceptible mice, in average, we have higher gadolinium infiltration in the brain, suggesting that the blood-brain barrier integrity is impaired or there's a loss of it. And so on the bottom right, you can see that we have a nice correlation that's significant between the level of gadolinium infiltrating the brain and the social interaction ratio. So the more loss of blood-brain barrier integrity, the less the mice will interact with each other. And so, like I said, gadolinium is pretty small. It's about 0.5 kilodalton. So we wanted to test how big the hole was in the blood-brain barrier. So we moved towards Evans blue. So Evans blue is going to bind to the albumin of the blood. So it's going to form a complex of about 65 kilodalton. So we inject it peripherally in the mice, and then we perfuse with PBS to remove it from the vessel. And so what we observed in the control with no stress, there was no infiltration of Evans blue. However, in the susceptible mice, we see an infiltration along the blood vessel that we do not see in resilient mice. So if we quantify that, again, we have a nice correlation. The higher infiltration of the Evans blue is correlated with lower social interaction ratio. So the more the blood-brain barrier integrity is lost, the more you're going to have social avoidance in these animals. And so we look for a mechanism to explain these findings. So we look at the different genes that are involved in blood-brain barrier integrity, and one that came out interesting is Claudin-5. So Claudin-5 is a tight junction, so you can see here it's going to close the gap between endothelial cell. It's contributing to maintain blood-brain barrier impermeability, and then it's changed in different conditions, a lot with inflammation related, like, such as ischemia, sclerosis, or glioblastoma. And so there's one mouse model that is knockout for this gene, and you can see at the bottom right that in a wild type mouse, if you inject a dye in the heart of the embryo, you're going to see the CNS is really well protected. But if you don't have Claudin-5, the gene is knocked down, then you can see infiltration of the dye throughout the CNS. And this is lethal for the mice. So after 10 hours after birth, the mice are going to unfortunately die. And so what we observe in our stress model is that you can see here the blood vessel stained with CD31 in red. Claudin-5 is kind of a long fiber along the blood vessel in green. And we see a loss of it in stress-susceptible animal. It becomes punctated, and there's kind of hole and little gaps here and there. In the resilient mice, it's not perfect like in the control, but it's looking much better. And so if we quantify the staining, we see again a nice correlation at the bottom right. So the higher the level of Claudin-5, the more the mice will interact with each other. So the higher the social interaction ratio. So having more Claudin-5 makes you more resilient to stress, basically. And so we checked if this was true in human as well. So we also confirmed that at the gene. This was protein with the microscopy. We also confirmed that with gene. And so we went to look into human tissue. So this is from 2 different cohorts. On the left, Montreal, Douglas Brain Bank, and then on the right with Carol Taminga, the Texas cohort. And so what we see at gene level is in people who commit suicide, were depressed at time of death. In black, in white you have the control, in black you have depressed without antidepressant treatment, and in red with antidepressant treatment. But all those people with MDD, major depressive disorder, unfortunately commit suicide. And you can see that we have this same loss of about 50% of Claudin-5 in both groups, and same for the Texas cohort. And so we went back in mice and check if we could rescue the Claudin-5 with an antidepressant treatment. So you have here imipramine, so chronic treatment, 35 days. Mice are screened for social interaction. We keep the control and the stress susceptible. Half of them receive a vehicle, the other half the antidepressant. And what's interesting is you can rescue with the antidepressant treated group the phenotype. So they're going to start interacting again. And you can see that it's also rescuing Claudin-5 expression. So this is one antidepressant. So in my lab, we're planning to test more different ones, including ketamine that I saw my colleague are going to talk about in the next few talks. And so in a nutshell, what we showed in this paper in 2017 is basically social stress induced a neurovascular pathology, including blood-brain barrier leakiness, which is going to promote depression. So you have on the left in resilient and control mice, you're going to have maintenance of blood-brain barrier integrity. So even if you have inflammatory mediators circulating, they're not going to cross the blood-brain barrier. And then you're going to have normal social and stress coping behavior. However, on the right, you're going to have inflammation, recruitment of immune cell, monocyte. They're going to release cytokines, including interleukin-6. And then those are going to affect the blood-brain barrier. It's going to become fragile. It's even going to open. And those cytokines we think could cross the blood-brain barrier and affect the cell on the other side. This is mediated by a loss of claudin-5 at gene level, protein level. It affects the morphology of the tight junction, and then leakiness of those circulating inflammatory mediator into the brain, leading to depression-like behavior, so social avoidance, the one I talked about. And so I won't go into details, but we also use viruses to modulate the expression of claudin, and we showed that it's in the paper that when you downregulate claudin-5 expression, you can also create depression-like behavior. So it's really a causal role of this blood-brain barrier leakiness. And so when I opened my lab, one question we had was, how do resilient mice maintain blood-brain barrier integrity? Because they are also exposed to the same stress. So how can they cope and do that? And so one thing we look into is epigenetics. So basically with epigenetics, you can have, it can be environment, it can be diet, it can be different life experience that are going to affect the gene expression. So we thought that maybe that could be a way that the resilient mice maintain their blood-brain barrier. So what we did is we looked what happened at the promoter of claudin-5, which will allow either repress the gene expression or allow expression of this gene that seems to be really important for stress response. And so on the left, you have acetylation, so which is permissive, and then it's always the same color scheme. So in white, you have the control, in black, the susceptible, and in red, the resilient mice. And you can see more acetylation, which is permissive to allow the genes to be expressed in resilient mice, but not in susceptible. And on the other side of the flip side of the coin, we see less methylation, which is repressive in the resilient mice again, and no change in susceptible. A bit like if the susceptible mice cannot cope or do not adapt their molecular machinery to the stress that they are exposed to. And so we decided to take a step back and look at the overall endothelium transcriptome. So what's going on in this endothelium, not only this specific gene, but all the genes that are related to the endothelium. So we use magnetic activated cell sorting, which is called MAX. So you see here, endothelial cell in the nucleus accumbens is going to be about 5–10% of the cell. So we enrich it using those magnetic beads, and then we can really see what's happening at the gene of the endothelial cell themselves. This is kind of a nice way to have a profile of a change in the endothelium. And so what we observe, for our surprise, there was a lot of change between control and susceptible, and resilient and susceptible, but we saw as many changes in control and resilient, which means that being exposed to stress and being resilient, it's not like being in unstressed control. You have some adaptation occurring in your blood-brain barrier. And so when we dig into what those genes are related to, you see here an heat map of all the changes. And the first pathway that came out is an inflammation-related pathway, so TNF-alpha and F-kappa B signaling. And so it suggests that the endothelium of the susceptible, if it's susceptible versus on stress control. So in susceptible mice, you have an endothelium that's inflamed. So it's going to affect a lot of different pathways. And then one gene that came out significantly different between the susceptible and the resilient is HDAC1, so histone deacetylase 1. So this is an enzyme that's going to be involved in epigenetic changes. So we thought it tied nicely with what I said for the first part, so the acetylation and methylation changes. So we dig a little bit more into that. And so we confirmed first that the HDAC1 change is also seen as in PCR, so quantitative PCR, not only in the sequencing. So you see about twice the increase of HDAC1 in our stress-susceptible mice, which is not observed in resilient animal. And again, if you do a correlation with social interaction ratio, the higher the HDAC1, the lower the social interaction. So the more the mice will avoid. So what we did is a pharmacological approach. So we infuse an HDAC1 inhibitor directly into the nucleus accumbens. This is always, again, nucleus accumbens, so it's region specific for the brain. So we did 10 days of vehicle or an MS-275 infusion, which is an inhibitor of HDAC1. And what we observed, it was the same paradigm as we did with the antidepressant treatment. So mice were screened for chronic social defeat, after chronic social defeat, kept the unstressed control and the susceptible, and then treated mice. And what we were able to do is reverse the phenotype of our stress susceptible animals, so they became resilient after the treatment, and it also rescued Claudin-5 expression. And so you have this nice correlation of the higher the social interaction ratio, the more Claudin-5 was expressed in these animals. And again, to wrap up and confirm what we've seen in mice and human, we went back to this cohort of patients. And so we saw a significant increase of HDAC1, like we saw in our stress susceptible mice, in MDD patients without antidepressant treatment at time of death, and a nice correlation between the level of HDAC1 and the level of Claudin-5. And we also confirmed by microscopy that in this human tissue, you also have these gaps in the tight junction of those people who unfortunately commit suicide and were depressed at time of death. And finally, we observe also epigenetic changes. And what's interesting is we see less methylation, which is again repressive, in people treated with antidepressants. So we think there's really an effect of the antidepressant on the blood-brain barrier. So this is something we again want to dig more into. So if I wrap up this second part of my talk, which is going to be kind of the end of it, you have the two papers at the top left. So the first paper, we showed that it induced neurovascular pathology. The second one, we kind of dig into the molecular adaptation, promoting stress resilience versus depression. So on the left, in those mice, you have acetylation on the Claudin-5 promoter, which is going to lead to normal social and stress coping behavior, because you kind of keep your level of Claudin-5 as in non-stress control, and then with that you are able to protect blood-brain barrier integrity. However, on the right side with the stress susceptible mice, you have inflammation in the blood, which leads to inflammation as well in the endothelium, activation of pathways, which will lead to change in epigenetics of the Claudin-5 promoter. You're going to have increased methylation, which is going to prevent expression of this Claudin-5. And then I didn't talk about it, but we also look at some transcription factors that are involved in Claudin-5 expression, mostly repression. One of them is beta-catenin and the other one FOXO1. So there's more detail in the paper. You have the title at the bottom. So it was published in PNAS earlier this year. And so what does it mean for you who's not a mouse? Again, this is always the big question. So at the very beginning, I talk about MRI. So we think that maybe by looking at the blood-brain barrier integrity, we could have a better idea of if people are depressed, are in recovery, or maybe diagnosis, especially with people who are more vulnerable. For example, people who had a stroke, maybe do a follow-up of psychological, how they feel about it as well. We think that there could be a potential for discovery of biomarker, because the blood-brain barrier has been a challenge to overcome for the drug industry. And so maybe if there's infiltration of circulating signal from the blood into the brain, maybe you could have something from the brain going into the blood that you could detect. And this way you could have a biomarker of depression that would be measurable in the blood, which would be a great advance. And finally, development of novel antidepressants. So instead of trying to cross the blood-brain barrier to act in the brain, maybe acting on the blood-brain barrier itself, reducing inflammation, could protect the neurons on the other side. And so we try to see the brain and depression and resilience and stress not only as neurons and brain related. Why? Because you can see here that there's been a lot of progress in several diseases in the last few decades. So with heart disease in light blue, you see there's been a lot of different new drugs that have been discovered, but unfortunately in mental health, depression in dark blue and schizophrenia in orange, it's like we're still in the 50s. So we need to come up with new ideas to try and new drugs to try to help those people. And so with that, I'm going to finish with, I have to thank my lab because it's not me pipetting anymore. And then so the work I presented was done by two very talented graduate students, Katarzyna Dudek, first author, and Laurence Dion-Albert, who's second author on the paper. My research associate lab manager, Manon Lebel, a visiting student from Ireland, Hélène Toc. Also, I have to thank the collaborators, Scott Russo, Gustavo Tsurecki, Naguib Meshawar, as well as the funding. And if we're going to have a discussion at the end, but if you have questions, comments, feel free to contact me. You have my email at the bottom. So with that, I'm going to pass the microphone, the virtual microphone, to Georgia, I think. Thank you.
Well, Carolyn, that was awesome. That's super exciting. And I know we talked about, I mean, if I have a bunch of questions. Why don't we just like have a few questions now?
Sure.
And yeah, we do plan for everyone watching at home. We do plan to have a discussion. Like I said, I think this is a really unique opportunity to get basic and clinical people together. I have a bunch of questions, but I'll hold mine and see if other panelists have questions. I think Georgia has a question. Yeah.
So Carolyn, that was fantastic. Given the sex differences in cardiac disease versus stroke between men and women, respectively, have you seen any sex differences in blood-brain barrier permeability or the effects of stress on such?
Yeah, so basically Laurent's project, that was a side project for her, but our main project is to do what I did when I was in New York with social defeat in male, but do it in females. And so what's very interesting and intriguing is she found differences in the different brain regions. So she doesn't see any change in the nucleus accumbens, but she sees changes in the hippocampus and prefrontal cortex. So we think that could explain or be part of why the symptoms of depression are different between men and women. But people often see vessel as being the same, but we think they are different. Also, they are bigger in the PFC versus capillaries in the neck. So clearly there's some sex differences. And this is something that we're digging into. And so she's going to do the same strategy I did. So inject viruses to open the blood-brain barrier in PFC in female and see what's going to happen with the behavior. But yeah, there's clear sex differences. As for stroke, this is something we want to pursue. I've been like submitting a grant about it because yes, there's also differences about that. And again, we think those sex-specific regional differences in vasculature response are a very intriguing finding and something we want to work on in the future.
And just as a follow-up on the, you talk about the regional differences, do you think that they're actually regional or they might be differences in timing? That you're getting, that maybe you're having a later infusion into prefrontal cortex and hippocampus or permeability that's clearing by the time you're measuring it in males versus in females?
Yeah, so where I'm at, the Cerveau Brain Research Center, my lab is in a neurophotonics center, so it's full of engineers and physicists who love to play with laser. And so what we're moving into is in vivo imaging. So we would like to have some, the mouse is going to have a little microscope, and then so we can test the blood-brain barrier integrity before the stress, throughout the stress, after the stress, but also maybe 1 month later, or have more like a better, because it's kind of the chicken and egg question. People ask me, do you think the blood-brain barrier was more fragile before the stress or it's caused by the stress? So I think it's going to be important to answer those questions and see if it's a long-lasting effect, especially because after 10 days we see epigenetic changes. So maybe, for example, if you do an early life stress, is the blood-brain barrier going to remember later in life? So this is another type of question we're going to try to answer. Thank you.
That's great. I guess the question I had to do with treatment resistance in humans, and very interested in your data showing that a conventional antidepressant, in this case imipramine, a chronic daily treatment actually reverses the pathology that you see at the level of the blood-brain barrier. So I just wanted your thoughts if, for example, have you looked, if you treat your defeated susceptible animals with imipramine, and then look at the effects on the blood-brain barrier, for example, as in addition to behavior. I think you reported, I can't remember exactly, but in general, an effective treatment versus a vehicle treatment, say. But is there a relationship between the rodents that have the, you may have presented this, where the behavior is rescued, basically where the antidepressant worked, versus I'm thinking about the animals that are defeated, they get treated with something like imipramine, they're still depressed. If you see that and starting to think about a model of treatment resistance in animals, and just clinically, we've always had this idea, and there's some data for it, but it's not a slam dunk, that inflammatory dysregulation in human depression in general, and maybe this question of a breakdown of the blood-brain barrier, do we expect that to be confined to individuals who are sort of treatment resistant to conventional antidepressants? I wanted your thoughts on that.
Yeah, so that's a really important question. So we think that there might be some subgroup of animals that the blood-brain barrier maybe has some factor that make it even more vulnerable to inflammation or other. Also, even the regional differences, again, so we are interested in the endothelial cell, but other cell types. So astrocyte, pericyte, the blood-brain barrier is composed of different cell types, and there's individual differences in human for that. So it's not as much study as for neurons, but also there's some like in the brain, there's some areas who are more permissive. The blood-brain barrier is less present, like choroid plexus, and so we don't know if, for example, the antidepressant treatment could leak into these areas and then after spread in the brain. I think it would be great to be able to inject the treatment and follow it kind of and see how it interacts and where it interacts in the brain. Again, we're moving toward having a whole brain imaging capacity. So not only looking at region we have an idea are going to be affected, like the nucleus accumbens, the PFC, but maybe looking at the whole brain. So using light sheet and clearing the whole brain and have an idea of the whole vasculature. Because yes, I think like in human and mice as well, that's why it's a good model. We have some subgroups. So, and it's not like a clear cut. You have a spectrum of animals, and then we see differences in the depression-like behavior. So you're gonna have some mice who are gonna be really social avoiding, but still drink sucrose, so showing no anhedonia and vice versa. But yeah, I think it's gonna be something interesting to explore in the future, especially in human with all the different subgroups.
Very cool. A lot to explore. I think in the interest of time, let's move on. If I could, but that was great. Thank you so much. Let me briefly introduce my friend and colleague, Dr. Georgia Hodes. I've known Georgia since she was also a postdoc, right, in Scott Russo's lab at Mount Sinai, quite a few years ago now. Interestingly, Georgia was one of very few of 6 researchers recruited to Virginia Tech to actually start the new School of Neuroscience. I believe that was in 2016. Many of you know her work. Her laboratory examines the biological mechanisms that contribute specifically to sex differences in mood disorders, and how those differences manifest in terms of immune mechanisms as well as neuroplasticity. She has over 50 publications, has had several awards, including 2 NARSAD Young Investigator Awards, and I'm very much looking forward to her talk. So with that, Georgia, let me hand it over to you.
And I just wanted to start out by thanking James and Carolyn for inviting me to be part of the symposium, and also thank you, SOBP, for putting this on virtually. It's really exciting to be a part of this. And I'm going to be talking to you today about some research I've been doing on microglial activation by stress and why sex matters. And I've been interested in microglial activation because there's been some indication in humans that this may be a biomarker of depression and particularly of suicidal ideation. So there are post-mortem studies that have shown that there's increased microglial density and activation in patients who have committed suicide. Additionally, recent advances in imaging have allowed us to image this in vivo in alive patients who have suicidal ideation or depression, and they show an increase in translocator protein. But it should be noted that this is not specific as a marker to microglial cells, and it also is activated in endothelial cells, which Carolyn has just so elegantly talked about. Additionally, in humans, antidepressant therapies are thought to inhibit inflammation, particularly by acting on a pro-inflammatory pathway in microglia, which we call M1 polarization or classical activation. And I'll talk a little bit more about that later. And what microglia are actually doing in humans and in mice is that they're involved in programmed cell death. So they're part of the normal healthy brain, and they're a homeostatic mechanism in which they engage in surveillance, in monitoring, and trophic support. They can be involved in, as I said, in programmed cell death and phagocytosis, but also in synaptic pruning and synaptic nibbling, where they're only eating components of the synapse, along with through their trophic factors affecting neuronal plasticity. When the brain is injured, we go through a period of pathological condition of inflammation, And inflammation is a normal part of the brain, but when it goes on too long and there's no return through homeostatic function, then we can get this aberrant microglial activation, which then involves things like excessive synaptic pruning, and this has been shown to be implicated in Alzheimer's disease, and in addition in obesity, where the microglia become less mobile, stay in one place, become activated, and also engage in this excessive synaptic pruning. And when I say they're activated, what I mean is that they're transitioning through different stages of morphology. So normally they're in this surveillant or quiescent state, also called ramified. But as they become activated, they become primed. And you can also think of this as being triggered or hyperramified, where now you have an increase in soma size and an increase in the number of processes and thickness of those processes, as they then move into this transitional state where they start to shrink in their processes and again grow a larger, more extended soma size till they're finally in this amoeboid form where they're highly mobile. And they can go back and forth between these different stages, but when we talk in the literature about being activated, generally we're talking about all of these different stages, so the prime, transitional, and amoeboid. And what we mean is that these are releasing certain kinds of cytokines, which are a messenger system in the immune system. And when we think about this microglial activation, one of the issues is that we can't tell the difference between this classical or M1 activation and this alternative or M1 or M2 activation, which have very different purposes. So this is a spectrum. Microglia exist across the spectrum. And in fact, the same microglia may be able to go from M1 activation to M2 activation. But when we think about M1 activation or pro-inflammatory responses, we're thinking about traditional inflammation. These cells are engaging in phagocytosis. They may be affecting blood-brain barrier permeability. They're involved in neuronal death and antigen presentation. Whereas with alternative activation, they're involved in inhibiting this inflammation. Again, phagocytosis, tissue remodeling through releasing trophic factors, and then restoring homeostasis. But what they're doing is very different, right? And they look exactly the same when you do immunohistochemistry, or most of the measures that we use for microglial activation. So they're being triggered by different receptors shown here, and then they're producing different types of cytokines. And what we know from the preclinical literature is that there are sex differences in microglial activation in response to stress, and that this seems to be really region specific. So work by Justin Bollinger in Kara Wellman's lab showed that acute restraint stress increased microglial activation, which was measured through these primed compared to ramified microglia in the prefrontal cortex. Whereas in females, he initially saw much higher levels of activation in just regular unstressed females, and this activation was lowered when these females were stressed. So there was an increase in the ramified or surveilling microglia in these females in the prefrontal cortex, and a decrease in these primed or what we consider partially activated microglia. At the same time, when we look at the hippocampus, there's reported to be an increase in IBA1 protein, which is a calcium indicator, which is a marker of microglia, for both the protein and mRNA in hippocampus in both males and females. And then recent work by Eric Walb with Ron Duman showed that when you exposed mice to chronic variable stress, we only saw increases in males in microglial activation markers. So in humans, we know that there are sex differences in the transcriptional expression of males and females who have major depressive disorder. And this was beautiful work done by Benoit Labonte when he was in Eric Nestler's lab, where he compared men and women with depression to same-sex controls and found very little overlap transcriptionally, particularly in a variety of areas involved in the prefrontal cortex. But he also found the same pattern in NAC, in the nucleus accumbens. And so in looking at microglial activation, knowing that there didn't seem to be a lot of effects in females in the prefrontal cortex, I wanted to look in the nucleus accumbens. And so what I used was a stress paradigm called variable stress, in which I expose male and female mice to a number of stressors. They differ by day. So it's foot shock, tail suspension, and restraint stress. And I can expose them for either 6 days or for 28 days. And then I look at their behavior starting either 24 hours after the last stressor or 30 days later. And I use these tests, such as splash test, novelty suppressed feeding, and forced swim, to look at different aspects of behavior. So in splash test, we have an ethologically relevant test where we're looking at a decrease in grooming. And anyone who's ever had a sick animal knows that they decrease in grooming, and in addition, in humans with depression, there's usually a decrease in self-care. In novelty suppressed feeding, mice are overcoming their fear of a novel environment to get a hedonic snack when they're food restricted. And in forced swim, we're looking at a measure of active versus passive coping to an environment. And I can then take all of the scores across these tests and generate z-scores and average them to get a stress susceptibility score. So what we're trying to do in this is to model the way that we measure depression in human beings, because we're looking at a bunch of different symptoms and how they relate to each other for a generalized score. And when we do this, if we look at their z-score or stress susceptibility score, and we start behavior 6 days after the stress, we only see behavioral effects in females and not in males. However, if we wait 30 days to measure behavior, we see effects in both males and females, suggesting that there's a stress incubation over time in males, even though these animals are group housed after the stressors. If we look at 28 days of variable stress, we get behavioral effects in both males and females. And again, this was long lasting. Up to 30 days later, we saw behavioral effects in these animals. When I had been a postdoc in Scott Russo's lab, we looked at the effects of 6 days of variable stress on transcriptional signatures in NAC. And here we were comparing stressed males to their same-sex controls and stressed females to their same-sex controls. And similar to Benoit Labante's study, we saw very little overlap between males and females exposed to stress. But we've been moving forward with the Bevilacqua group to now reanalyze some of these data using CompBio. So we're looking at the relationship of these genes to each other, and particularly what themes are being expressed or targeted. And what we found in this reanalysis is that in females that are stressed for 6 days, we see this really large upregulation of immune regulation and tolerance. So this seemed to actually be the biggest impact in terms of a system based on the genes that were regulated. And in fact, when we looked at what was downregulated in females versus males, it was reduced immune function. So it suggests that females are at a heightened immune state when they undergo 6 days of variable stress. So we started out by just looking at whether males or females had more microglia in their nucleus accumbens. And we divided this into shell and core to get a better understanding of what was happening in the nucleus accumbens. And we were somewhat underwhelmed to find out that males had more microglia than females in the shell. And there was a transient decrease in microglia in females after 6 days of stress, again in the core, but not in the shell. So, this wasn't particularly informative, so we moved on to look at the morphology. And when we looked at the morphology, we started to see real differences. So first and foremost, we only even saw the amoeboid microglia in females after they were exposed to 6 or 28 days of stress in both shell and core. We also got an increase in these primed microglia and a decrease in these surveillant microglia, which was in comparison with males, where we saw either no effects or, if anything, the opposite, where we were getting a decrease in the prime microglia at 28 days of stress when males are stress susceptible, and an increase in their surveilling or homeostatic microglia. So it suggests that there's a very different pattern of activation of microglia in females versus males, both after 6 and 28 days of stress. And in particular, when we looked at that ratio of primed to surveilling microglia, we saw these effects very strongly in females in both core and shell, most predominantly following 6 days of stress, but still significantly increased in females after 28 days of stress compared to their same-sex controls. So we were excited to see this activation of microglia in the females and thought that this may be involved in some of the sex-specific effects we see, particularly at 6 days of stress. So we next asked if we could block the effects of stress on behavior in females through inhibiting this M1 classical activation. And we did this in part because this has been shown to be antidepressant in male animals. And so we gave our mice minocycline in their drinking water for 14 days and throughout behavioral testing. And we asked this question of, if we inhibit an M1 activation, is this antidepressant? And the short answer is no. It's really not antidepressant in females. And if anything, in animals that are unstressed, it promoted a stress-susceptible behavior. So in females that were not exposed to an exogenous stressor, they behaved like they had been stressed when they were just given minocycline in their water and then given these behavioral tests. And so our first thought was, well, maybe in females, this is not reducing activation of microglia. And so we went on to check that. We've looked at the 40 mg per kg dose. And what I am showing you here is that stress is increasing the number of processes and total branch length in females, supporting the idea that they are primed with stress. And it is decreasing this when they're stressed and given minocycline. But interestingly, when they're not stressed in minocycline, it's actually increasing this. And so one of the things that happens with minocycline is it's an M1 inhibitor, which means it may be switching these microglia over to an M2 activation. And so we are now looking at this question of M2 activation and whether that's what's actually promoting stress susceptibility in these females, or an interaction between M1 and M2 status. In particular, I know it seems counterintuitive. Sorry, John, I need to stop for a second. I know it seems like this would be counterintuitive, that M2 or anti-inflammatory activation would be involved in any kind of detrimental effect on behavior. But in the body, it's this M2 activation that's actually involved in allergies. This is still an immune response, which leads us to fight an invading pathogen, which in this case would be parasitic worms, right? So this has an inflammatory purpose, and it may be that in mistaking stress, these animals, or inflammation or minocycline, these animals are having an aberrant immune response. We've been trying to understand what this microglial activation might do, and particularly put it in the context of some of the work that we've done before. This was beautiful work that was done by Dan Christoffel when he was a graduate student in Scott Russo's lab. And he showed that the pathway from the thalamus to the nucleus accumbens was what specifically produced social stress susceptibility. So in this case, he activated glutamatergic inputs from the thalamus to the nucleus accumbens and was able to drive stress susceptibility in male mice. And when he looked at glutamatergic projections from the medial prefrontal cortex to the nucleus accumbens, he found that those were actually involved in resilience. And he had initially come upon this idea by looking at vesicular glutamate transporter 1 and 2. So VGLUT1 is going to be expressed from projections from areas like the prefrontal cortex and the hippocampus to the nucleus accumbens. And he didn't see any differences between animals who had gone through social defeat who were either resilient or stress susceptible in VGLUT1 expression, nor did he see a relationship to their social interaction ratio. But when he looked at animals who had gone through social defeat stress, he saw an increase in VGLUT2 puncta coming from the thalamus and brainstem. And again, this correlated with their subsequent behavior from social defeat stress. And so he proposed that this projection would then promote stress susceptibility. And working with Anna Brancato, who was a visiting graduate student to Scott Russo's lab, we found that we also saw an increase in VGLUT2 in females after stress, particularly after the 6 days of stress. But interestingly, we also saw a decrease in VGLUT1. So this was different from what Dr. Christoffel had found in the males who were stress susceptible, that he was not seeing a change in VGLUT1, where we were seeing a decrease in these females who had gone through 6 days of variable stress. And so one of the things we're looking at now is whether microglia are eating VGLUT1, if they're pruning this presynaptic function particularly. And so what I'm showing you here is a microglia stained for IBA1. The arrows are pointing to VGLUT1 in red, which is being engulfed by that microglia, and we can determine that by looking at CD68 staining, which is a glycoprotein found in lysosomes, to show that the VGLUT1 is actually in the lysosome. And so we're wondering if there's a preferential engulfing of VGLUT1 in these females after 6 days of stress. And so to try and bring this all together, others have reported that in females, there's either an increase in these homeostatic surveilling microglia in the medial prefrontal cortex and a decrease in priming that's occurring. And at the same time, we're showing you that there's this increase in priming in the nucleus accumbens in females and a decrease in these homeostatic surveillant microglia. And so what we think might be happening is that there's a mechanism through microglial activation in the nucleus accumbens that's reducing this resilience pathway from the prefrontal cortex. Whereas in males, this may be happening actually in the prefrontal cortex itself by an increase in microglial priming. And in closing, I've shown you today that there are region-specific effects of stress on microglial activation that are specific to sex. John, I'm going to go back for a minute. I have to move this. All right. In conclusion, what I have shown you today is there are region-specific effects of stress on microglial activation that differ by sex. That in females, M1 inhibition does not block the effects of stress on behavior in the absence of an exogenous stress, actually produces behavioral stress susceptibility. And this is counter to what has been previously reported in males. And that microglial activation in the nucleus accumbens may contribute to a presynaptic neuronal change that we know is involved in stress susceptibility. But what's really important about these data is that they show that we need to investigate sex-specific biomarkers and treatments. If we are going to look at things like microglial activation in humans as a biomarker, we need to understand if it's going to be the same in the regions of males, of the brain of males and females, and whether if we target this with novel antidepressants, if that's actually going to work in females, because they are the predominant sufferers of depression. And in closing, I want to thank the members of my lab, in particular my graduate student, Mariah Seglova, who did the majority of this work, along with Emily Hurst, who is an undergraduate who helped her immensely. And from the Russo lab, I'd like to recognize the work of Dan Christoffel and Anna Brancato and our funding, and I'm happy to answer questions.
Thank you, Georgia, for a wonderful talk. What a treat to be able to see that type of mechanistic detail on microglia and the role in stress susceptibility and effects of sex. Really interesting. I have a quick question, and then maybe we could open it up to some of the others. You referenced at the beginning of your talk some of the human data with the translocator protein. It's been very interesting. It's also been discussed and debated how to interpret it for many of the reasons you said. I don't recall, but you may have mentioned it. Do you know, have investigators that have used that method in humans looked at effects of sex and brain region.
So they've looked at brain region, but most of what I've seen has been cortical, and they have not looked at sex, nor have any of the human postmortem studies looked at sex as a variable. So they haven't segregated by sex. They'll use samples from both men and women, but in particular, one of the issues is that a lot of these samples are from people who committed suicide. And as you know, while women are more likely to attempt suicide, men are much more likely to complete. So a lot of the suicide banks are very heavily skewed towards men.
Okay. So a lot more work to be done there. Just another point in terms of translation, you mentioned minocycline, which likewise has attracted a lot of interest. There's been some clinical trials. We've dabbled in that area a little bit in bipolar depression. The evidence of antidepressant effects of minocycline in humans is mixed. So it was really interesting, again, trying to think how human and translational studies would be informed by some of these detailed biological mechanisms. If I understood you right, you did find, as expected, that minocycline reduced the classical M1 activation. Was that in males and females, or can you just remind us?
We've only specifically looked so far in females. We are looking at males at these measures as well. With minocycline, one of the things that was most interesting to me translationally was when I got this very surprising effect that we had an effect of minocycline actually promoting stress susceptible behavior in the females. I went through the literature. There are very few reports in animals of minocycline in females. I think there may be one paper that's been published. And when I talked to other people, they said, oh, because it doesn't work in females. And then no one's published this. And
that's called a publication bias.
Yeah. And then I couldn't find anything in humans on its relationship, on whether a subset of people get depressed when they take it. But if you look through some of the Reddit threads, there's actually quite a few on people who become suicidal when taking minocycline.
Really?
And again, I think, it wasn't clear cut as to whether this is a sex difference or an individual difference, but I think this is something really important that we need to look at and understand a little bit better.
Great, super interesting. Other questions by panelists?
Me. I have a question. Great talk, Georgia. I really like this work. I was wondering when the microglia goes from surveillance mode to like the more angry mode. I cannot pronounce the last step. I'm sorry. Can they come back? Like, do you see it as in the stress condition? They just stay really angry, or they go back and forth like between surveillance and very phagocytic?
Right. So one of the ideas is that they are going back and forth. At least it's unclear whether once they become amoeboid, whether they'll transition back through the other stages, but definitely through the primed and transitional, they can go back to being surveillant. And one of the things we want to do is use nanoparticles to really track microglia and see if we can see these changes in morphology and whether the same microglia are staying activated, if it's the same cells or if they're changing back to being surveilling.
And so my second question was, I don't know about if there's that many studies about sex differences in microglia, just basal gene expression. I was wondering if there's, for example, for the synaptic pruning, is there some gene that are more expressed in female versus male, and then it would be easier to prime, or in different region?
Yeah, so Stacy Bilbo has done some really beautiful work in her group looking at these baseline sex differences actually throughout development. Particularly in the early stages, there do seem to be quite a lot of sex differences. It's been less clear at baseline whether there are sex differences in adulthood, and a lot of the research suggests that it's not as pronounced at that point as it is in development when they're really engaging very actively in the synaptic pruning. But one of the ideas is that stress may reawaken this sort of earlier activation and engagement of microglia that you see in development. that's using developmental mechanisms.
That's great. Thank you again, Georgia. So I think now we're at the halfway point. We're right on time. I think we're going to switch now to the human half of the symposium. And Carolyn, I think you're going to take over kind of moderating for us.
So it is my great pleasure to introduce the next speaker. So Dr. James Murrough, who's going to talk to us about dynamic peripheral immune profiles and neurocircuit response to reward in patients with depression. So James is an associate professor of psychiatry and neuroscience and director of the Depression and Anxiety Center for Discovery and Treatment at the Icahn School of Medicine at Mount Sinai in New York. He is a faculty member of the Friedman Brain Institute and Center of Excellence in Neuropharmacology at Mount Sinai. Dr. Murrough conducts clinical and translational research aimed at understanding the biological basis of mood and anxiety disorder in order to point the way towards new, more effective treatment. So his lab has received funding from the NIH, the Doris Duke Charitable Foundation, American Foundation for Suicide Prevention, the Dana Foundation, and the Brain and Behavior Research Foundation. So I pass to you, James, the virtual microphone.
Thank you, Caroline. Much appreciated. And like I said at the beginning, it's a real treat to be together with this group, reviewing some really exciting biology from basic and clinical perspectives. So let me jump right in. My talk today is dynamic peripheral immune profiles and neurocircuit responses in depression. That's a human depression. These are my disclosures. I do consulting for industry as listed there. Some research support that was a number of years ago. A few patents that are filed. Nothing, no royalties. I'm going to briefly mention ketamine. There is a patent that's I'm not named on it, but it's at Mount Sinai that's related to the use of ketamine and S-ketamine for depression. And just in terms of more detail about this institutional conflict with ketamine, you can see here it involves Dr. Charney and others at Mount Sinai. Okay, let's see. Okay, so depression is the largest cause of disability worldwide. This has been a number of years now, but essentially this is our largest disease worldwide. In terms of morbidity and disability. It's been estimated to cause over 70 million years lost of disability. And we've already heard about suicide several times today. Obviously, it's at the top of everyone's mind in mental health treatment and research, trying to stem the tide of suicide, which to date, unfortunately, as a field, we have not done. There's more than 1 million suicides every year worldwide, and the trend is actually increasing a bit rather than decreasing. So we have a lot of work to do. It's estimated that up to 1 in 3 patients with depression, unfortunately, don't have a good response to our current treatment. So that's kind of why we're all here. Just in terms of, this is an older slide, but in terms of trying to think about what we might call the clinical heterogeneity of depression. This is a nice just graphic I like from Calabrese. Really sort of clinically, we know this. You know, I do think that what we call depression in humans, it does tend to hang together in kind of a grouping of a constellation of symptoms. And you can see this artist has demonstrated this here. Some of them that we commonly see aren't even in the classic DSM description of depression. For example, you see the sort of anxiety and irritability cluster. And then there are other ones, and what I'll be focusing on today is you see down in the lower right, sort of the anhedonia, closely related amotivation, and fatigue. But, you know, why? I mean, as a field, we've been sort of moving to try to focus our research efforts on these clusters or aspects of heterogeneity. Why? Well, we expect that these symptom clusters or symptoms, we'll be able to map them to more specific psychobiological constructs, if you will. We think that in the future, they will anchor future nosological or diagnostic systems, and presumably they'll be fruitful targets for future therapeutics. This is all still empirically to be determined, but this is just sort of guiding the way we think. And obviously everybody knows the sort of NIMH version of moving this forward that's known as RDoC, or research domain criteria, is sort of a one organized and structured way of approaching this more general a problem or goal of breaking apart the clinical heterogeneity of these complex disorders such as depression or mood disorders into more tractable constructs. So with that, you know, the talk today, I'm going to be telling you about some recent work we've done in depression, but for this project and for many of our studies, we have endeavored to more specifically assay at the level of clinical symptomatology, brain circuitry, and other measures, these specific domains, if you will. So we'll be talking mostly about anhedonia today. I've given you the DSM definition. It didn't change from 4 to 5. As often with the DSM, it's a little bit clunky, but it reads, so anhedonia is the markedly diminished, or the symptom of depression that we sort of call anhedonia in DSM is markedly diminished interest or pleasure in all or almost all activities most of the day, nearly every day. More simply put, we might refer to anhedonia as the reduced capacity to feel or perhaps respond to pleasure. And, you know, I'll mention, although I won't go into it in detail in this talk, anhedonia itself is a broad construct within depression as a broader construct. And there are many aspects to anhedonia that may map to different stages in sort of the processing of reward, for example, anticipation, consumption, et cetera. It is one of 2 required symptoms to make the diagnosis of a major depressive episode. And we know from epidemiologic studies, for example, that the presence of greater levels of anhedonia are associated with a number of poor prognostic factors, such as a chronic course of illness. It does tend to be associated with treatment resistance, poor general functioning, and has also been linked specifically to suicide risk above and beyond general severity of depression. It's attractive. I won't get into sort of the transdiagnostic aspects in this talk. We have focused on depression, but presumably it's a highly transdiagnostic construct across mood disorders, including bipolar, and even more broadly, obviously, in terms of psychotic disorders. So it is an attractive target from sort of a transdiagnostic RDoC perspective. I mentioned, you know, in general, we can envision anhedonia as arising from specific aspects of deficits in reward processing. This is just one on the left. I've listed just 4 types of reward deficits and the way they might be measured in humans. This is based on a review that I cite in the lower right corner. We can think of deficits in consumption, our consumatory reward. So that's you might think of sort of the experience of pleasure after consuming a potential reward. And that is generally measured with a scale called the SHAPS. We could also think about what's probably quite important, anticipatory reward. So that's the ability to anticipate and have your behavior guided by the anticipation of getting a reward. And that's classically measured with the anticipatory scale of the TEPS, which is the temporal experience scale. And there are other ones that I'll be speaking less about today, such as motivation, that can be measured with what's called the effort task, and also reward learning that I will talk about that is measured in several ways, but including the probabilistic reward task. And I'll be presenting a little bit of data on this. This was developed by Diego Pizzagalli, and it's sort of a classic task, computerized task to measure implicit reward learning. And briefly, what you see in the lower right, this is sort of a classic finding. You compute response bias, and essentially the individuals interacting with the computers with 2 stimuli that are very similar, and they get feedback that says you won a small amount of money. Trial by trial, they have to pick between stimuli that are very similar. It's an implicit task. One of the stimuli is known as the rich, and it's reinforced more. Long story short, healthy individuals develop what's called a response bias. They tend to pick the rich stimulus trial by trial, block by block, on average more than the lean stimulus over time. And you see that in the red bars in the indicated as the controls on the lower left. This is from a paper from one of the early papers from Dr. Bevilacqua and a more recent review he wrote, where you see the response bias compared to depression in the blue, where as the trial moves forward, it's relatively attenuated. So I mention this because this is sort of a classic objective measure of deficits in reward learning that has been implicated in depression. Moving to the brain on the right, this is a nice depiction. There's been, as many of you are well aware, very exquisite detailed work in rodents and primates, in humans with structural and functional imaging, detailing a reward circuit essentially with key components that we've already been hearing a lot about today, including the nucleus accumbens and its projections to the medial prefrontal cortex. Okay, so I told you about anhedonia. That's kind of the clinical lens through which we're viewing this problem, at least for the purposes of the study I'll tell you about today. Switching back to the immune system, you've been hearing a lot about it. Fortunately, my colleagues have done a lot of the legwork for me. But just very briefly, at a high level, to review what are sort of the reasons why we think the immune system is implicated in depression. Well, it starts with that classically, it's observed that a significant proportion of individuals who don't otherwise have depression, but they're treated with the pro-inflammatory interferon alpha for hepatitis C, develop depression. And that was sort of the classic observation. We're still trying to understand how that all works. We know that patients that have a primary immune disorder, such as rheumatoid arthritis, have an increased risk for depression, presumably above and beyond the impairment that can be attributed to the primary immune disorder. I'll show a little bit of data that it does seem that immunological treatments in patients with primary immune disorders show amelioration of their depression. And there are now multiple meta-analyses in patients with primary depression that show small but consistent elevations in circulating pro-inflammatory cytokines. And I've listed a few here, classically interleukin-6, TNF-alpha, IL-1 beta, and others. And this was touched on, I think, a little bit earlier in the conversation, but the evidence for the efficacy of anti-inflammatory treatments in depression, I would say, is mixed. It's not my intention to review all that here. Certainly we can talk more about that. So this is classic work by Georgia when she was in Scott's lab. You've heard about the paradigm, the social defeat paradigm, but I wanted to show this because sort of was motivated the human study that we did that I'm going to show you. So just to remind everyone, in her work on the top left, so this is in the context of the social defeat model you've heard about, where following 10 days of daily defeat, animals can be segregated into what's called susceptible, or sort of a pro-depressive, pro-anhedonic phenotype, and resilient. So what you're seeing in the panel A is you're seeing circulating levels of interleukin-6 immediately pre-defeat, where you see they're essentially minimally different, but then immediately after defeat, you have a large surge in IL-6 that is much larger in the susceptible animals compared to the resilient. I'm showing you on the right, this kind of got us, we had been banking samples from patients that were in our studies or coming through our depression center. And Georgia said, well, why don't we take a look at those? So you see there, in fact, we did find a number of years ago, consistent with others, that, for example, IL-6 was higher in our patients with depression versus control. Really interestingly, I want to point your direct attention to the lower left, because what I'll be telling you about is a new study where we've taken the blood cells, the leukocytes from patients, and done this ex vivo stimulation. And this was why, because in Georgia's study, this is ex vivo stimulation with lipopolysaccharide in the animals before they ever went through the defeat, which showed that the animals that were susceptible had a large increase in IL-6 upon stimulation, but the basal levels were not different. So you can see that in panel C, and likewise in panel D, the greater the LPS triggered IL-6 increase, the more of a depressed phenotype with the SI ratio that we heard about previously. And just finally, a little bit of data on the right showing that in fact, giving a monoclonal antibody against IL-6 can rescue the phenotype as well as knock down circulating levels of IL-6. So one study we did based on some data that we had available is these were in patients that were receiving ketamine as part of a clinical trial. This was published by Corrali et al. A few years ago. This was, I would say that the take-home message was we were a little bit underwhelmed at the power of basal circulating levels of cytokines to tell us a whole lot. What you see at the top left, these are healthy controls and patients with, in this case, they had treatment-resistant depression because they were eligible to be in a clinical trial of intravenous ketamine for depression. Here you see small but statistical increases in IL-6. Here, what you see in panel A, B, C, D, these are just the four sort of canonical, most commonly, this was not a multiplex approach, but most commonly observed, it was multiplex, but we focused on the top four, and of the top four that had been reported in the literature, actually only IL-6 was significant. The bottom half, when we asked the question, well, what happened with ketamine? And what was the association between change in peripheral cytokine levels and treatment with ketamine? Again, it was fairly underwhelming. What you see in panel E is immediately 4 hours after a single infusion, there's a small reduction in IL-6 that was significant 4 hours later, but not in most of the other measures. And when you look 24 hours later, that's in panel F, the bottom right, there's essentially no difference. And importantly, we couldn't correlate changes here with their antidepressant response. So this sort of taught us that it was probably going to be relatively limited how far we could move forward with this approach. Before I show you the new data, this is just some of the data I had mentioned before, published a few years ago, again, focused on IL-6, where this analysis was pooling two randomized controlled trials looking at two separate anti-IL-6 therapies for, on one hand, rheumatoid arthritis, and another immune disorder called multicentric Castleman's disease. And what's being reported here, and the authors were asking, if you look at the subgroup of patients that had these primary immune disorders that had depression and anxiety based on the SF-36, and then you asked whether those, it changed with treatment, the answer was yes. So at the top, this is the sirukumab trial in patients with RA. At the bottom is the ciltuximab trial, both anti-IL-6 therapies. And you can see that there's an improvement. Here, confusingly larger scores is less depressed and less anhedonic on the scale. Importantly, of course, when you actually adjust for their improvement in their immunologic conditions, in that case, actually, you can see that it's still significant, but only for ciltuximab. But after adjustment, it's no longer significant for sirukumab. So in general, there's some evidence, but it's not a slam dunk. But armed with that information, we received a small grant from the NIH to try to see if by stimulating leukocytes from patients with depression, we could sort of uncover some more interesting details. So what were the goals of this study? Our goals were to characterize, on one hand, the basal, again, circulating levels using a larger multiplex protein assay. We also wanted to characterize the dynamic release of leukocytes. And we wanted to specifically try to relate these changes to measures of anhedonia and reward processing. Our approach was to do ex vivo stimulation, as I mentioned, with LPS, essentially following the protocol that's been done in animals, to use functional MRI with a variant of the monetary delay task that classically activates the reward system, and to use the probabilistic reward task that I introduced earlier. And this study was led by Drs. Costi and Morris in the lab. At the bottom of the screen, you see kind of the flow of the study. It was only 2 visits. There was a screening day where individuals go through informed consent. They go through a thorough diagnostic and medical history, a physical exam, screening labs with any clinical, to rule out anything, any clinical problems. And then following all that, they come back for their assessment day, which begins with questionnaires in the morning, a blood draw in the morning with plasma and PBMC isolation, and then moving on to the brain and the behavioral reward tasks. This is the sample. So we had 51 individuals in the sample of 30 depression and 21 were healthy controls. You can see the ages, late 30s. Interestingly, George, you might be interested in this. So this is showing proportion of male. As expected, the depression group is predominantly female, although not hugely so. But actually, interesting, our healthy group is more male. And for those reasons, we did adjust for sex in the analyses, but I think there's more to unpack there. If you go to the bottom, we assess their general depression severity at baseline using the MADRS, which goes from 0 to 60, with high 20s or around 30 being a moderate to moderately severe depressed. And then the SHAPS, which has sort of been a classic general measure of anhedonia, tends to be thought to be more about consumption. There's a lot of questions that say, if, how much would you enjoy, how much pleasure would you receive from engaging in one or another activity? And they give some examples, a cup of coffee, flower, things like that, smelling the flowers. Interestingly, it's been a little bit unclear in the field what would constitute clinically significant anhedonia. Some have said in the field it's as low as 20. In general, anyways, you see our depression group had about a 35, and our healthy group had just over 17. In this scale, higher scores is more anhedonic. So just very briefly, this is the ex vivo stimulation procedure, fairly straightforward. We did this in collaboration with our human immune core. And really what we ended up with what was called PBMCs. So when I say leukocytes, it's a mix. We think it's mostly dominated by monocytes, but we purified PBMC, and then it was incubated for 6 hours with LPS as indicated there. And we used the Olink platform to do the immune assay. So what did we find? So this is kind of the raw data. These are the top hits. We had initially, we looked at 92 analytes, some were undetectable, et cetera. So, but when we cleaned the data, we were actually able to use imputation techniques to recover. These were the top hits. If you Bonferroni correct, you don't really get significance. That's probably not the right way to do it, because these are not, if you to Bonferroni correct, you would have to think that each immune pathway was in independent observation. But what you see here is this is fold change for each analyte in the stimulated versus unstimulated condition, and we're plotting the healthy controls now in Mount Sinai colors, pink, and light blue for depression. What I don't say on this slide, which I should, is when you look at the unstimulated plasma levels, which is what, the first observations that we had with IL-6, others, that was not different between groups. So we went into this experiment thinking that the stimulated levels should reveal group differences if, based on the hypothesis that leukocytes, maybe monocytes, we're not sure which cell type, is sort of dynamically hypersensitive in depression. Our findings somewhat support that in the sense that actually the unstimulated levels were not different by group. Here's our old friend IL-6 and some other common ones, and ones I had to look up. So what do we do with this data? Well, so we thought, all right, what we wanted to do was we took the fold change data and we simply entered it into a principal component analysis for data reduction. And what you're seeing here is on the left, you're seeing the essentially the raw data now just expressed as a difference of fold change. Okay, so TNF came out at the very top. You can see also the chemoattractant ligand 20, IL-6 at the top, coming down on the left-hand side. We took the fold change data, entered into a PCA, and basically the first component explained over 50% of the data. It included the factor loadings, as I indicated there on the right. And when we simply do a t-test of PC1 score between the groups, which we thought was the most parsimonious way of doing it, it confirms that there's a larger inflammatory response in depression versus healthy, which is indicated on the bottom right. So now we wanted to try to link this with symptoms. So what you're seeing here is, I mentioned, so we did adjust all analyses for age, sex, and BMI. We were interested primarily in using the SHAPS, but also the anticipatory scale of the TEPS. And what we found on both scales was evidence supportive of our hypothesis. Interestingly, the strongest association was with the anticipatory score. And I'll show you some data. We think that so far, based on what we've seen and what's in the literature, we think that hyperimmune signaling might be specifically related to the anticipatory components of aspect of reward, although that has to be more rigorously tested, obviously. But here we got our largest association with the anticipatory scale of the TEPS. So what I'm showing you on the right is that association where this is across everybody now. Now, just to confuse us, I've put MDD in red, sorry about that, healthy in blue. And you can see, so with the TEPS, I have to clarify, a higher score is better. A higher score is more experience of the anticipation of pleasure. And as expected, you have sort of a cluster of the healthies congregating on the right-hand side of the x-axis, whereas depression is on the left. And you see the association there with the immune score. So moving to the brain, just briefly, we used a task called the incentive flanker task. This is in collaboration with Dr. Emily Stern, who's a colleague at Mount Sinai, who's now at NKI and NYU. Essentially, it's a variant of the monetary incentive delay task. There are 3 trial types: a reward, loss, and neutral. And trial by trial, individuals are told that they could either receive or even lose a reward in the form of money if they perform the task correctly, which is displayed in the diagram of the task on the left. And then they get response that says, correct, you win. And you can model in this task either the anticipation or the consumption of reward. Anticipation is actually easier because anticipation is basically the point at which, so if you see it's indicated as X, so they're told, and that's on the screen for a few seconds, they're told on the next trial you could win if you do well. So what we're modeling is the brain state in the expectancy condition versus the neutral condition. I don't think my pointer works, so I won't bother to try, but it's sort of a gain over neutral, which is classically the way the MID has been analyzed. On the right, taking the lead from our friends you've already been hearing about, of course, we're interested in the nucleus accumbens. Apologies, I may interchange that with ventral striatum or VS throughout the talk if I haven't already. I see it on the axis there in panel B. We used a region of interest approach based on all the data you've been hearing about today. When we extract the key contrast of interest, which is gain over neutral within the nucleus accumbens, that data is plotted on the right as a function of group. And as has been previously shown, we show attenuated activation to reward in the depressed group versus healthy. But our real goal was, one of our main goals is to understand, was the activation of the NAC to reward related to the peripheral excitability of the leukocytes? So we were very pleased to find that, in fact, it was. So this, to me, is probably the most exciting finding of the study. The text you see there on the right is actually, that's just general additional information about how we approached the functional MRI analysis. I just didn't have room on the prior slide. But in terms of the finding, which is the graphic on the left, there again, you see the principal component 1 score, which is sort of our data reduced single value for immune activation, if you wish, versus anticipation to reward. And what you can see is the higher the immune score, the more impaired the activation, and the scale goes from -10 to 10. So we were very excited about this. Okay, let me just move on to how am I doing on time? Let me move on to the probabilistic reward task that I mentioned earlier in the talk. Implemented this in collaboration with Dr. Pizzagami and Alexis Witten. So just briefly, I sort of already explained this, but it's a signal detection task that provides an objective assessment of reward learning. And you can see there, I sort of explained the process of the task, choosing implicitly between rich and lean stimuli, where a response bias or reward learning is computed. So in this study, we were able to replicate a prior work, which is shown on the right, that as expected, depressed patients have a blunted response to reward, or a reduced, what's called reward bias. Of course, we were interested in two things: whether this was associated with their self-reported anhedonia levels. Well, three things: anhedonia levels, the immune markers, and the brain. The strongest association was actually with the brain, interestingly. So in line with the hypothesis, the greater the response bias, so the more healthy or more normal, if you will, was associated with greater activation of the nucleus accumbens to reward. So that was good to see. The association with the immune activation was less certain. So there's more to be done here. There was a trend in the right direction. So you see that there's an R value of -0.23. So a small, but it's in the anticipated direction, where greater immune activation score reduced reward learning. But that did not reach statistical significance with p.16. And if I have it here, I don't have it here, but there was actually essentially no relationship between self-report, interestingly, and this task. So still questions, but I think for me moves us a little bit towards trying to put these mechanisms together and link peripheral and central. So let me just conclude as follows. So what I've talked today, hopefully I've conveyed that anhedonia is an important characteristic of depression, and it seems to arise from deficits in reward processing in the brain we might be able to measure and understand. As you've been hearing all morning, converging lines of evidence implicate immune dysregulation in depression, and arguably linked specifically to anhedonia, although I didn't really get into that literature particularly. We think we found that circulating leukocytes are hyperreactive to an immune challenge in patients with depression versus healthy volunteers when controlling for age, sex, and BMI. And I've shown you that greater peripheral leukocyte reactivity is associated with self-reported deficits in the experience of anticipatory pleasure. It's also associated with blunted reward response to anticipation in the nucleus accumbens. And so this is just the beginning, obviously, but clearly much more research is required to understand the biology of leukocyte dysfunction in depression and the processes that would link this peripheral dysregulation to central mechanisms. And obviously we've already heard some hints about how that might happen, how the periphery and central systems might be in communication via changes in the blood-brain barrier. So this is our lab at Mount Sinai. I want to thank all the funding. The study I just presented was funded by the National Institutes of Mental Health. The study was led by Doctors Sarah Costi and Laurel Morris. This was done in heavy collaboration with Scott Russo's lab, and inspiration from some of the earlier work from Georgia that I mentioned, and external collaborators, including Diego, Alexis, and Emily Stern. So with that, I'll stop there.
Great, thank you, James. That was a terrific talk. I'm going to start with a brief question, if it's okay. I was wondering if there was any in the panels you did inflammatory marker or mediator that surprised you, that you found. Like you look at like the classical one, but was there one that was really surprising?
So yes, there was more than one. And I mean, honestly, we and I are still trying to understand and try to chase down what some of these pathways, what the implication would be. There's something called tweak that looks really interesting. But what I found is that the biology of many of these pathways are not fully understood, and certainly their role in mood disorders. So what I found was basically I had a bunch of new analytes that to look at, and for future studies, try to think of ways, whether it's through a gene expression or other mechanisms, to try to drill down on a number of pathways that were absolutely not on our radar before this study.
Awesome. So any other question? Georgia, did you want? Yes, go for it.
Oh, great talk, James. And I'm so excited to see this work, honestly, especially the PBMC stimulation. I was wondering if you've done any work using flow cytometry to identify what cell populations are actually in that blood, because a lot of the markers that you have coming up seem to indicate a push towards increases in the innate immune system, particularly monocytes and possibly neutrophil attraction.
Yeah, so great question. So we're doing that now. The way we collected the blood, I think doesn't allow optimal cell sorting and identification of subpopulations, which Scott is well aware of. And we have a new collection protocol now where we're doing that sorting and also preserving the cells so we can wake them up and do stimulation or other approaches in the future. So I think we suspect that there are absolutely specific cell populations that are contributing to this signal. So that's going to be an important part of the next, I think, project.
And then just sort of following up, when you think about anhedonia and depression, while there's a decrease in pleasure, different forms of depression, when you think about atypical versus melancholic, for example, are engaged in eating more or eating less. And I was wondering if you tracked any of that in your sample and if that might change some of the cytokine responses.
That's a great question. So we did track it because I may have skipped over this, but all the participants underwent a SCID, so sort of a structured diagnostic interview, which does allow us to define the subtype of the depressive episode as melancholic atypical. So we have that data, and I don't think we've looked at that. My guess is the sample is going to be too small. In most studies in general, trying to link the classic depressive subtypes have not been fruitful, but we should definitely look at that. And I think what we're going to find is many patients, because they do have a mix of, they have some melancholic and some atypical features, they often don't end up with an episode specifier. It'll say like unspecified, or so often it's sort of, you would want it to be informative, but often it's sort of not. So we use the dimensional measurement anhedonia, but that's a great question. We could also check, whether SHAPS or TEPS scores associate with these more classic subtypes of depression. I think that's a good point. Could look at that.
So it is my great pleasure to present the last speaker of our symposium, so Dr. Laura Bevilacqua. She's going to talk to us about gene expression and molecular pathway associated with rapid antidepressant response to ketamine in patients with treatment-resistant depression. So Dr. Bevilacqua is assistant professor in the Depression and Anxiety Center for Discovery and Treatment at Mount Sinai, New York. There's kind of a trend here with Mount Sinai. Laura completed her medical training and obtained a PhD in the neurobiology of affective disorder at the University of Pisa in Italy, and subsequently joined the Laboratory of Human Neurogenetics at the NIH, where she studied functional genomics and gene-environment interaction relevant to impulsivity and aggression. So her current work at Mount Sinai, she's conducting translational research towards the understanding of the neurobiology of affective disorder and the development of novel therapeutics. So I pass to you, Laura, the virtual microphone.
Thank you so much. Thank you for having me today. I've learned a lot from these terrific talks, and thank you for giving me the opportunity of presenting this study that we just recently concluded. So yes, I'm going to present on the gene expression and molecular pathway associated with rapid antidepressant response to ketamine in patients with treatment-resistant depression. Now you've heard already a lot about major depression, how common it is. It's about 6% of the adult population worldwide is affected each year. It's a leading cause of disability. You already heard a lot about the association right between depression and chronic inflammatory conditions, such as diabetes, cardiovascular disease, which increase the burden of the disease. There is a 20-fold increase in risk for suicide compared to the general population. And the response to conventional monoaminergic antidepressant is usually delayed. It takes several weeks. And 30% of patients who suffer with depression do not remit. Their symptoms do not remit even after several treatment attempts. These individuals will meet the definition of like treatment-resistant depression. And despite the clinical relevance, little is known about the mechanisms of TRD. Now, recently, it has been shown, including by Dr. Murrough, who did some of the pivotal studies, that a single administration of low-dose ketamine had a rapid and sustained antidepressant effect. And that ketamine is clinically effective in patients who have failed to respond to conventional monoaminergic antidepressant. And recently, last year, was approved by the FDA in the form of S-ketamine for the treatment of treatment-resistant depression. Ketamine is an NMDA receptor antagonist, but the mechanisms of action is complex, and it's unclear how it ultimately leads to its antidepressant response. So we've heard again a lot about immune system and depression, and this sort of like bidirectional relationship between the peripheral immune system and the brain, and how they might interact in causing depression. We have already heard that from the previous presenters, that a subset of patients with MDD, right, with major depression, show chronic low-grade inflammation. With increased inflammatory cytokines in the blood. We've seen increased, been reported increase in IL-6, TNF-alpha, IL-1 beta are the mostly reported. Changes in peripheral cytokine levels are also associated with antidepressant treatment response. And pro-inflammatory state is associated with treatment resistance. For instance, James earlier talked about the Kirali study showing that there is an increase in IL-6 at baseline in patients with treatment-resistant depression, although this was not associated with treatment response. So what I wanted to do what we wanted to do in this study was to investigate the whole blood transcriptional profile related to treatment-resistant depression and gene expression changes that were associated with treatment response to ketamine. And so to do so, we compared whole blood transcriptional profiles between healthy controls and patients with treatment-resistant depression at baseline prior to receiving ketamine. And then we looked at pre-existing transcriptional differences between ketamine treatment responders and non-responders, so at baseline prior to treatment with ketamine. And then we looked at gene expression signatures associated with clinical improvement, measured by a change in depression symptom severity at 24 hours after ketamine treatment. So a little bit more in detail, we screened 21 healthy controls and 26 treatment-resistant depression patients. And they all underwent standardized screening at the depression and anxiety centers. Both the healthy control and the treatment-resistant depression patients were free of medications known to affect the immune system, and the patients with depression were free of antidepressant medication. Furthermore, they were healthy, did not have like any chronic inflammatory disease, any autoimmune disease, and were not currently sick with any like systemic inflammatory disorder. For healthy controls, we collected blood samples at baseline, and for patients with TRD, we collected blood samples immediately prior a single ketamine infusion and at 24 hours after treatment. We collected peripheral whole blood in PAC-stream tubes for RNA isolation and sequencing. The efficacy outcome was at 24 hours after the single ketamine infusion, and depressive symptoms were measured with the Montgomery-Åsberg Depression Rating Scale. Ketamine responders were defined as having at least a 50% or more reduction in a MADRS total score at 24 hours, and non-responders compared to their baseline, and non-responders were defined as having less of a 50% reduction in symptoms measured with the MADRS. The healthy controls and patients at baseline did not differ in age, gender, and BMI. We had 18 out of the 26 TRD patients, 18 were ketamine responders at 24 hours, while 8 were non-responders. And also responders and non-responders did not differ in social, demographic, and clinical characteristics. Okay, so we then looked at gene expression, differential gene expression patterns between TRD patients and healthy controls at baseline. And all the models for gene expression analysis were adjusted for age, for gender, for BMI, ethnicity, and for RNA integrity number. And the significance was set at a p-value of 0.05, and a fold change threshold was set at 1.3. So here what we see is on the left a volcano plot showing the over 19,000 express gene, and in red and in blue are the genes that are respectively significantly upregulated and downregulated in patients with TRD compared to healthy controls. The genes that are labeled are the top 25 genes differentially regulated between the two groups. The significantly differential express genes were then fed into the Ingenuity pathway analyzer software to identify pathways, gene pathways, underwent also, we also did a gene ontology analysis. And what was so interesting is that we had interferon signaling pathways and interferon type 1 signaling pathways being sort of like our top result. IPA, the Ingenuity pathway analysis software, also can predict upstream regulators. And in TRD, compared to healthy controls at baseline, we had an increase in expression, we had a predicted upstream regulator activation of genes in the interferon signaling pathways. And we've heard already about interferon signaling pathway in depression from the previous presenters. Interferon, now a long time ago, patients, it was observed that patients with hepatitis C treated with interferon alpha developed susceptibility to develop depressive symptoms. And this finding has sparked sort of like the interest in in immune system modulation and depression. A few years ago, there was a paper from Mustafavi et al. That also looked at whole blood gene expression in major depressive disorder. This was not a sample of treatment-resistant depression, but of major depression. And they also, their top finding was that the interferon signaling pathway was activated also in their sample. And interestingly, there's an 80% overlap of interferon pathway associated genes between our study and that study. And this is important because we've been able to sort of like replicate the importance of this pathway, but it also might indicate that our sample, which is relatively small, may be representative also of a larger MDD population. At a baseline in the patients with depression, we also did a weighted gene co-expression network analysis, which is a method used to sort of identify groups of genes that show similar expression patterns. And here we can see how the genes cluster together, and we identify 11 modules. And we then correlated the modules with clinical characteristics of depression in our population. And the genes that were associated with certain clinical characteristics, like age of onset or recurrence of illness, for instance, are all modules that contain genes that play a role in immune regulation. We then looked at differential gene expression patterns at baseline, so before ketamine administration, in ketamine responders and ketamine non-responders. So again, we did IPA analysis and identified a CMP mediated signaling pathway, a neuropathic pain signaling pathway. And interestingly, these are the genes that were differentially expressed within these pathways. And what is we thought very interesting was that we found two glutamatergic genes, the GRM2 and the GRIN2D gene. Now we know that ketamine is a glutamatergic system modulator, and it's been hypothesized that ketamine might cause a glutamatergic surge and an activation of AMPA receptors that will lead to increased synaptic activity. Now, GRM2 and GRIN2D are glutamatergic receptors. The first one is a group 2 metabotropic glutamate receptor, and it's mainly expressed. I borrowed this image from Zanos et al. It's a review of possible mechanisms of ketamine in depression. But so the GRM2, the metabotropic glutamate receptor 2 is mainly expressed presynaptically in proximity to presynaptic terminals, and they act as an outer receptor, and they can decrease synaptic glutamate release. And it is possible that ketamine, by antagonizing this receptor, would disinhibit the blockade of presynaptic glutamate release and allow for like a glutamatergic neurotransmission and glutamatergic surge. The GRIN2D receptor is a glutamate ionotropic receptor, NMDA types of unit 2D, and ketamine has high affinity for the 2D subunits of the NMDA receptors, and these are mainly expressed in GABA up here on the right, and GABAergic interneurons. And inhibition of GABAergic interneurons may lead to to pyramidal cell disinhibition and therefore enhancement of glutamatergic neurotransmission. Of note, also these receptors are expressed peripherally on leukocytes, and so we could also, potentially hypothesize that ketamine might have an action of these receptors also in the periphery, and maybe modulate inflammatory, modulate the immune system, inflammatory responses, inflammatory states in depression via peripheral action. Finally, we looked at the treatment response to ketamine. So we included in our model different changes in MADRS scores per patient. So we looked at change in MADRS score, so in depression severity scores after treatment with ketamine compared to baseline. And what was very interesting but in this analysis was that we observed an inhibition of predicted upstream regulators after ketamine response compared to activation of those same upstream regulators at baseline in TRD versus healthy controls. Finally, we also looked at genes that were upregulated and downregulated, individual genes that were upregulated and downregulated in TRD versus healthy control at baseline, and that were up and downregulated by ketamine, so after treatment. And we identified 16 genes. And we were drawn in particular to IDO1, so to endolamine 2,3 dioxygenase 1 gene, because of it, and we'll talk about it again in a second, but because it could be a possible link between immune activation and glutamatergic abnormalities. So IDO1 was upregulated in the TRD patients compared to the healthy at baseline, and the change in IDO1 expression inversely correlated with the change in MADRS, meaning that a downregulation of the gene correlated with improvement in MADRS scores after treatment with ketamine. And again, the IDO1 gene encodes for IDO, so the endolamine dioxygenase, which is a tryptophan catabolizing enzyme that is activated by inflammation, and that once activated might lead to the generation of glutamate-like compounds, such as quinolinic acid, which can alter glutamatergic neurotransmission. And so, to summarize our findings, this is, at least to our knowledge, the first study utilizing an unbiased transcriptomic approach to investigate the rapid antidepressant effects of ketamine on whole blood gene expression in a sample of treatment-resistant depression patients, which are medication-free. We saw that patients with TRD compared to healthy controls are characterized by activation of interferon signaling pathways at baseline. We also saw that prior to ketamine administration, the GRM2 and the GRAIN2D genes are enriched in ketamine responders versus non-responders, indicating that these receptors, again, could play a role in the response to ketamine, and that treatment response to ketamine is associated with a downregulation of several genes coding for inflammatory proteins and interferon signaling, and it's associated with a downregulation of the IDO1 gene. And so thank you very much for listening. I want to thank all the Depression and Anxiety Center team, and in particular, Molly Schneider, who was one of the research coordinators of the study, of course, James, and Dennis Charney, and the Scott Russo lab, and in particular, in Florin Katomas, who I worked very, very closely with, and the Eric Nestor lab and the Li Shen lab, who all helped out tremendously with the project. Thank you.
Great, thank you. That was another terrific clinical talk. I love clinical talk. It's really different from what I'm doing. I have a question that might be a naive one. So I was wondering, when you have those non-responder, are people who are non-responder to ketamine going to be non-responder forever? Or can you move from non-responder to someone who's eventually going to respond? Because I was wondering if you could see different gene changes in those people.
So sorry, say it again. So it's non-responders to ketamine?
Yes, I was wondering, if you are a non-responder initially to ketamine treatment, can you become a responder eventually? Or are you going to be a non-responder forever?
So, so there is some, and maybe James can talk more about this, but there's some evidence from clinical practice that some patient might actually not respond immediately to ketamine, but might respond after a few doses, sort of a few treatments of ketamine. I mean, the mechanism, I don't think we know what the mechanisms of that are, or how to predict. We can't predict who's going to respond at a first.
I think that would be amazing to have the blood of someone who's not responding at the beginning and eventually respond, and maybe you will have some immune adaptation, or you know.
Yeah, that's a great thought. Thank you.
Georgia, did you want something to add about? Do you want to add something about that?
I mean, I just thought it was also really interesting that one of the top pathways besides interferons was mitochondria. Mm-hmm. That, you know, this idea that perhaps there's a difference in energy regulation of the cells themselves might be contributing to depression. When you think about sort of the macro and the micro, it's a nice relationship to think about.
Yes, actually, we also did an MDC, a module differential connectivity analysis using the WGCNA identified modules. We then sort of like looked at how the sort of like the relationship between the connectivity between genes in each model would change in depressed patients compared to healthy controls, and there were two modules that were significant, and one of which was a large module containing many genes, a few I think it was around 4,000 genes, but the top hit pathway was mitochondrial mitochondrial regulatory pathways. Yes.
Great. So if we don't have any more questions, James, did you want to move on to a wrap up or discussion between panel?
Yeah, no, thank you. Yeah, no, this has been great. And I think, you know, I know we're running a little bit long, and I think we just sort of spontaneously were taking advantage of having interactions between talks, which probably is better, because by the time 2 hours goes by, no one's going to remember what the questions were. So I think we kind of I was really happy. I think we had a nice, robust interaction between the speakers as we went. And since we're over time, I don't think we need to have a big wrap-up session. So if everyone's okay with that, we'll just sort of wrap up here. Really want to thank the speakers. This was super interesting to me. Really happy the way this came together. You know, this is the type of research we love. You know, Georgia and Caroline, everyone, you know this is what we like to do. Try to hear what you all are doing to give us ideas, and hopefully hearing what we're doing is interesting the other way too. So I'm just really glad. Thanks again to the Society of Biological Psychiatry. It's always been very supportive, and I'm sure I've gone to more Society of Biological Psychiatry meetings in my career than in any other research meetings. It's just, I think, the perfect place for this. type of interaction. So Carolyn, glad to co-chair with you, and we'll just say thanks to everyone, and hopefully the viewers will find this interesting as well.